at least to some of you who doesn't know her. This is Valentina Boeva. She is a team leader in Gauchamp Institute.
I don't know how to pronounce it in English. In French it's Gauchamp. I think it must be Gauchamp or something like this.
Yeah. From Paris, and yes, she started her group this year. She focuses on computational genetics and epigenetics of cancer. She knows almost everything about cancer, and she will try to tell you, share some part of her knowledge today. So please.
Yeah, thank you. I will try to speak loud so that I don't need a microphone, but if you don't hear me, you can always ask. Alright, so I wanted to talk to you today about the epigenetic states in normal cells and how these states can change in a cancer cell and why cancer cell wants to change them. I will start with the normal cells. So, you know that you know what's chromatin, right? You know that DNA is wrapped around nucleosomes, and if you take DNA from one cell and stretch it out, you will have 2 meters. While a cell is very compacted because DNA is wrapped around protein complexes, nucleosomes, and in addition to the function for protein compaction, these nucleosomes they can have chemical modifications that define chromatin states, and these chromatin states they make part of epigenetic states. When we speak about epigenetics, or when I speak about epigenetics, I mean several things. I mean modification of DNA, CpG manipulation. I mean modification of histone tails. I mean use of histone variants. And I don't mean microRNAs, many things. But I mean also 3D structure of chromatin. So I will speak a little bit about all this. First for normal cells, and then I will mention how this can change in cancer. Alright, so I will start with DNA methylation, then I will show you a couple of slides about histone variants and histone modifications. Then I will introduce to you hidden Markov models and how you can use this technique to decode the chromatin states using information from histone modifications. I will talk a little bit about techniques to find open chromatin regions and to study the 3D structure of the genome, but I think this topic has been covered yesterday by Katerina. And then I will end up by changes of epigenetics in cancer. Alright, so DNA methylation, you know that CpG can be methylated on cytosine, and if you see this modification in the promoter region, then most likely the gene will be silent. Because these modifications will develop chromatin status, but also in some cases they can prevent transcription factor binding. So some transcription factors can bind only on unmethylated regions. How we can detect these regions, I think it has been covered too, so I'm going quickly on this. You can use the bisulfite treatment of DNA, and when we use this bisulfite, then all cytosines which were not methylated, they become uracils, and when we sequence them, we see T instead of C. So then you can, instead of sequencing, actually you can use also DNA methylation arrays produced by Illumina, also works pretty well. And then you can visualize the data in the UCSC genome browser. And here, for example, you have the CpG methylation profiles for 2 gene regions. If you have methylation, you will see this region in orange. If you don't have it in blue. And for example, when I look at this gene, SPIV, in the promoter, I see that in these 2 cell lines, it's blue, meaning it's unmethylated. And in these 3 lines it's orange, meaning it's methylated. So I presume that this gene is expressed only in this first and the last cell line because the promoter region is not methylated. Alright. This is one type of epigenetic modification. Another type of epigenetic modifications, histone modifications and histone variants. You know that nucleosome is made of 8 proteins. 2 pairs of each canonical histone So we have histone 3, histone 4, 2 like this, and histone 2A, histone 2B, 2 like this. 8 in total. But instead of using these canonical forms of histones, in some places of chromatin, we use, the cell uses histone variants. And there are quite many of them. This is for human genome. Usually, a histone variant differs from the canonical histone by just several nucleotides, several amino acids. Sometimes they can be longer or shorter. The difference is really minor. And I must say that usually histone variants are expressed at much lower levels than canonical histones because they're not used throughout the genome, but just in some particular places. For example, we know that this variant of histone 3, which is called 7PA, usually binds centromeric regions and help inside chromosomes chromatin segregation. Then 3.3 can be found in promoter regions. Histone H3AZ, again, it can be found in promoter regions because it opens the helps to open chromatin and so on. Alright, so we were speaking for histone variants. Now I'm going to talk about histone modifications. So we take normal canonical histone, and this histone has a tail, and like this. And these tails can be chemically modified. And actually they can be phosphorylated, acetylated, simulated, many different modifications can be applied. So here we have this histone 3 and all possible modifications you can have on the tail of histone 3. And interestingly, these modifications are associated with particular chromatin or transcriptional states. For example, if we look at the trimethylation of lysine 4 of histone 3, if we see this mark somewhere in the genome, we think that this must be a promoter. It can be an active promoter, or it can be poised promoter, meaning a promoter that can become active very quickly. If we see trimethylation of lysine 27 of the same histone 3 on the promoter region, we know that this gene is silent. And we know that the mechanism of silencing of this gene is by polycomb repressive complexes. And many marks are associated with different gene activation or gene repression, and also distribution of these marks around transcription start site or in the gene are different. So here I show you distribution of 6 different histone marks around the transcription start site. So on the left you see region downstream of the TSS minus 30 kb, and on the right side you see region upstream. This is upstream, sorry. On the left is upstream TSS, and here is downstream the TSS. And I show different genes in different expression by different colors. Highly expressed genes are shown in red, and silent genes are shown in purple. And this is intermediate expression levels. And you see that, for example, for this repressive mark, which is put on by polycomb complexes, only silent genes have this mark around transcription start site. No expressed genes have this mark after the TSS. Almost the same goes for H3K9 trimethylation, which is a mark of hydrochromatin. Again, all the silent genes have this mark, much less of them, but still, no active genes have this mark. If you look at this one, H3K27, 36 trimethylation, you see that this mark is present in actively transcribed genes, and then it starts at the TSS and then goes to the gene body. And this is actually a mark of transcriptional elongation. If you see this mark, you know you are in a gene body and this gene is actively transcribed. If you look at this mark, H3K9 acetylation, you see it only on the TSS, and this is actually a mark for active enhancers or active promoters. It's actually more or less the same distribution as you have that for H3K27 acetylation. So using these histone marks, you can predict really well the level of gene expression. So here it's a figure from this paper from Ann Consortium. On the x-axis, you see predicted gene expression values in log Q scale, and on the y-axis, you see measured gene expression values using KH for the technique we've just talked about. And we see that using these histone marks, we can predict gene expression level really well. Not only we can say gene is on and off, we can say the exact level of expression of this gene. So no need to do RNA-seq or KH anymore, just do ChIP-seq.
Well, the other way around, ChIP-seq is much more expensive.
It was a joke. All right. So to see these marks, you do ChIP-seq. If you don't know how ChIP-seq works, I will walk you through the main steps. So what you want to know where a particular protein binds to DNA. And this particular protein can be, for example, a histone variant with H3K27 translation on its tail. So what you do, you cross-link DNA with proteins, so that proteins cannot unbind from DNA, they are fixed. Then you cut this DNA using, for example, ultrasound into small pieces. Then you take an antibody which is specific for your protein of interest, for example, H3K27 translation. And then you extract DNA protein complexes using this antibody. You reverse cross-links to get pure DNA. And you do site selections because you want to have DNA fragments always the same length, about, for example, 300 base pairs. And after this step, you have a library of DNA fragments. Each DNA fragment was bound by a protein of interest somewhere. And your fragments, they make 300 base pairs, plus minus. And then each fragment is sequenced from one end. You have a read. You map this read to the reference genome. And if you see a cluster of reads, you know that your protein was binding somewhere. Usually, when we analyze ChIP-seq data, we don't work with reads. We work with the densities of signal. And to get this density, we need to do a read extension back to the length of initial DNA fragments. So we take these mapped reads. We extend each read to the length of the initial fragment. And we calculate density, which is just the number of fragments that cover each position. And we get the signal like this. The higher the signal, the higher the probability that our protein was sitting there. And then we can visualize the signal, which is a big file like this, in a UCSC genome browser or EG. So you will see signal along the genome. And actually, by looking at the shape of the signal, sometimes you can even say where nucleosomes were sitting, because the signal comes from nucleosomes. It's the nucleosomes that have tails, they shall modify. So for you, if you work with ChIP-seq data, I would advise to use EGV because you don't need to load all this data somewhere. It takes much less time, less heavy. However, if you use UCSC, you can use lots of public information, so it can be also good. Yeah.
Do you do read extension for not for transcription factors?
Yes.
For histones?
Yes, of course, you do.
Why do you do that? Because read extension means that you think your protein is sitting in the middle of the region? In the middle of the read? No, in the middle of the fragment.
Actually, you don't. Even for transcription factor, you don't think so. What you think?
Do then?
You think that your protein was sitting somewhere along this fragment. You have a fragment, and your protein can be here, there, there, there. But just probabilistically, when you have lots of fragments covering this position, when you calculate this density of coverage by fragments, you will see that the density is highest where your protein was sitting.
Yeah.
So, but there is no difference between a transcription factor or a histone.
Okay.
It's just, one is the protein, another was the protein. So I don't see why you're using different techniques.
I was thinking about broad peaks.
Here you can have broad peaks, but it doesn't matter. You can still extend your read to the length of the fragment, get this density.
Okay, thank you.
Can I add a little bit? Because the comment about broad peaks is relevant, but it's mainly relevant when you use a peak finder afterwards to identify where do you have a peak and where do you not have a peak. Then the properties of the peak finder will be slightly different depending on whether you're looking for broad peaks or narrow peaks. But for mapping the reads onto the genome, it does not really matter.
And so this is the story about mapping.
Yeah,
Okay.
and the next story about finding the peaks
I see.
computation.
I see the distinction.
All right, thank you very much. So what I wanted to say is that using, I don't know why I don't have it. I have it here, but not there.
Click one more.
Sorry? Yeah, okay, good. All right, so what I want to say is that when you have all these histone modification signals along the genome, you can define chromatin states. By chromatin states, I mean active promoter, poised promoter, active regulatory region, transcribed gene, and so on. So, for example, if in the region you see this H3K36 trimethylation mark, you know that you're in a region which is gene body, which is transcribed. If, in addition to the signal, or actually independent from the signal, you have H3K27 acetylation plus H3K9 acetylation plus H3K4 monomethylation, you know that you are in an enhanced region because also in this region you don't see H3K4 trimethyl, which is a marker for promoters. Because if you just have H3K27 acetylation, it can be promoter or enhancer. But if it's a promoter, you would see this mark. If you don't see it, it's an enhancer. In addition, you know that we are in a gene body here. So you can do segmentation of the genome using these histone modification marks. And different teams propose different number of states you can use to do this segmentation. In ENCODE, they propose 7 states. You need enhancer, promoters, CTCF binding sites. Since later I will talk a little bit about it later, and so on. Another team, Manolis Kellis and his group, they propose 51 states, but they are grouped. So, for example, here you have 6 promoter states, then several gene body, transcribed gene body states, and so on. And if you look for each state, so states are here, it's 51 states here, and this is marks. You will see that it may be that just there is a small variant that can describe the different states. So, for example, all these states are promoter states, and they are characterized by this promoter mark H3K4 trimethyl. It's just one here has, in addition to H3K4 trimethyl, it has H3K27 trimethylation, which is a repressive polycomb mark. And this state will be a repressed promoter, also called bivalent promoter. How many states to select when you want to do this segmentation? My answer would be use your biological intuition. Although the Manolis Kellis and group, they said use the following method. Score each model based on the low likelihood of the model minus a penalization on the model complexity determined by the range and information criterion of 1/2 of number of parameters times the natural log of the number of intervals, and this gives you 51 states. For me, there's three kinds of lies, deadlines, and statistics. So this is for me an application of statistics to give reason to your biological intuition. All right. So now I wanted to tell you how you can use hidden Markov models to segment the genome into these states. This is because I want to talk to you about bioinformatics. Otherwise, my talk will be pure biology. It's not maybe the purpose here. So, what's hidden Markov model? I will start with the Markov chain. In Markov chain, you have states. For example, here you have 3 states. One state is rainy day, another one sunny day, and then there's a state which is called eternal night. And you have probabilities that connect states together. You can go from rainy day to sunny day with about 30% probability and so on. And then at some point, if you go to the eternal night state, you stay there forever. The probability to go back is 0. Yeah. And if you observe in these states, if you observe that this is this Markov chain, then when you want to speak about Markov model, things become more complicated. In addition to the states, you have observations. Yeah? Okay. For example, you can observe what I do. You can look at me, and if I can go shopping, walking, breathing, and I can sleep. example, on a rainy day, I would rather shop. Sunny day, I would rather walk. Also, when it's raining, I prefer to read and so on. And then, internal times, I only sleep. But imagine you don't see the states. You have only observations, the second level. So I go shopping, walking, and so on. And then I sleep for many, many days. Apparently, you can guess what's going on, right? So this is a hidden Markov model. You have only observations, but you would like to guess the states. So this is formal definition. So states can change in time, but we don't observe them. States define observations. And if you use the first order HMM, then state at the moment t plus 1 depends only on the previous one. And we would like to decode states when we have observations and we know about probabilities to go from one state to another, and also it's called transition probabilities. Or we also have emission probabilities, which is probability to have some observation given the state. When we apply to our chromatin data, our states are this promoter, active enhancer, transcribed gene states, and observations are histone marks. So, for example, this is the states, 7 states, voice promoter, active promoter, so on. You have transition probability to go from one state to another when you just go along the genome. So, for example, if you are in an active promoter, you can go to a transcribed region, or you can go only to the transcribed region, right?
But you can stay in that promoter state.
Oh yeah, you can stay for some time, and then you can go to the transcribed region. If you're in the transcribed region, you can stay in the transcribed region for some time, but then you can go to transcription and from there to silence chromatin, for example, or somewhere else. I think I made this drawing, so I kind of make some mistakes. Anyway, so you have these transition parameters, probabilities to go from one state to another. In addition, you need the emission probabilities. If you are, for example, in a transcribed state, what's the probability to have this mark here, also very high, or have this mark also high, and have this regression mark as very low. And we have this emission probability for each state. Summarized like this. And then you can decode your chromatin states using this method, the algorithm. And it has been implemented in ChromaGenM developed by analytical telescope. So you can use it if you want. Also, in the ENCODE, you can download these states from major cell types. All right. If you are rich, you can do ChIP-seq for many marks. Sometimes we don't have lots of money to do ChIP-seq, but we also would like to have some information about chromatin states. For example, about open chromatin, which is binding sites of transcription factors, promoters, and so on. And in this case, we use 1 to 3 experimental techniques: FAIRSEq, Dynase hypersensitivity sites, assays, or MXC. All these techniques will give us information about open chromatin, which is correlated with these marks and anti-correlated with this repression mark. I will just show you in the UCC genome browser how this profile for Dynase hypersensitivity sites can look like. This is along the genome. So this is the profile. Here you see that there are many transcription factor binding sites where you have peaks in this profile. And if you look at these two other tracks, you will see that this one is like a sum of these two. Here you have binding sites of CTCF, transcription factor, and here you have H3K27 acetylation. So this is a mark of active enhancers and promoters, and these are insulators. So CTCF is a protein which is used to create loops of chromatin, and I think you should have heard about it. Yeah? Yesterday, yeah, exactly. So what you can see, if you do this open chromatin assay, you will get information about active promoters and enhancers plus CTCF binding sites. Alright. And if you do another type of experiment at ATAC-seq, you get exactly the same information. So DNAs and ATAC-seq, they give you the same result, although the techniques are slightly different. So DNA is hypersensitivity. You just add DNAs that cuts DNA where chromatin is open. So the more reads come from regions which is open chromatin because you had more cuts there. And ATAC-seq is for transposase assay. So this is a transposase that can put adapters where chromatin is open, and then you use these adapters to amplify these regions. But the effect is absolutely the same. The advantage of ATAC-seq over other techniques is that you don't need much material. It works really well when you have only 50,000 cells, and even you can go single cell. So here I show you profiles for 254 single cells for ATAC-seq. So you can really look at each cell what region is closed, what region is open. Although the resolution is not perfect, but if you combine things together, you can figure out. Okay, so I'm going really fast. So I would like now to tell you a little bit about the 3D organization of chromatin, because it's also related to epigenetics. Have you heard about lamina-associated domains, LADs? I see yes and no. Good. So you know that chromatin is like this in the nucleus, and some regions are attached to the nuclear lamina. They're called LADs. And in these regions, there are less genes. So you see that in the LAD we have less genes, and in regions which are not attached to the nuclear lamina. And also genes that are located in LADs are not really, they're silent. So it can be a way for a cell to silence genes. You just put them close to the nuclear lamina, and they're there, nobody sees them, they're not expressed. RNA polymerase II doesn't bind them. To see these regions, lamina-associated domains, we use a special technique which is called DMID. I'm not going into details of this. If you want, you can read it. Yeah. But in the end, if you use this DMID technique, you can again see these lamina-associated domains at the single cell resolution. So here you have different cells, 6 individual cells, and this is the profile averaged over 100 cells. You see that the signal is really very pure. So you can, for each cell, you can see whether chromatin is attached to lamina here or not. In addition to LADs, you have TADs, topological isochain domains. There is a very good correspondence between LADs and TADs, because there are TADs which are attached to lamina, and they are LADs. At the boundaries of TADs, we have a CTCF binding like this. Yeah. So we have TAD, CTCF, another TAD. And often, if you look at the histone marks, we will see that one TAD is enriched in one specific mark. So this one here is enriched in H3K27 trimethylation, meaning that this is a TAD which is repressed. And this mark, H3K9 dimethylation, is actually a mark for LAD, for lamina-associated domains. And again, you see that this TAD is enriched in this mark. So you know not only this TAD is repressed by polycomb, but you know that this TAD is attached to the nuclear lamina. And in cancer, all this can be changed, but this is I will describe it in the end of my talk. Using information about TADs that you obtain using high-C or full-C or high-C techniques, you can try to reconstruct the shape of your chromatin. And there are several physical models to do it. You can refer to this publication if you want to know more about it. I told you that the boundaries of TATs are limited by the CCCF binding, and you can have, in a normal cell you can have this structure, 3 TATs, CCCF binding here and there. This TAT is repressed, this one is repressed, this one is active. However, if you just mutate this CCCF site, 1 point mutation, these 2 TATs can fuse, and then you don't know what will happen. So in this drawing, this repression domain was extended, and we observed gene silencing. In some other cells, an enhancer which is located in this type can start regulating promoters from the other type. So we can observe different types of dysregulation if we mutate these TAT boundaries, CTCF binding sites. Yes?
Do I understand it correctly that these TATs, these 2 TATs are rats? Yes.
Not every TAT is a LAT.
Yeah, but these 2 that are repressed,
They may be LATs. But to know if they're LATs or not, you need to do H3K9 dimethylation.
Yeah.
If you see it, you can say it must be LAT.
okay, thanks.
Lots of information about LAT structure, TAT structure, histone mark, histone variant distribution can be found in ENCODE for many cell lines, normal cell lines, cancer cell lines. So here's just the top ones. And by arrows, I show cancer cell lines. And you can have RNA-seq data, SharePad data, many things. So if you want to study chromatin or just play around with this information, just go to ENCODE. All right, so this was for normal cells, and now in the remaining time I will talk about cancer. The questions I try to answer is whether epigenetic states change in cancer cells compared to normal states, and whether there are some phenomena which are specific to cancer cells, something that strange is going on there. And indeed, there must be something on the epigenetic level. For example, here I show you the list of genes which are frequently mutated in different cancers and that code for chromatin remodeling genes, epigenetic writers, readers, and erasers of histone modifications. So these are proteins that can read histone modifications, that can put these histone modifications, or erase them. And here you see proteins that are related to DNA methylation or demethylation, like TEC2. TEC2 is a protein that puts hydroxymethylation, actually replaces methylation by hydroxymethylation, and it's super frequently mutated in different cancers. You can see EZH2 mutated in many cancers, and EZH2 is the main component of the polycomb repressive complex 2 that puts H3K27 trimethylation, repressive mark. You can see mutations in MLL proteins that methylate DNA and lysine 4, and this is an active mark, and so on. So the fact that we see so many mutations in cancer in this type of genes tells us that we must observe genome-wide epigenetic changes in cancer cells. For example, we can observe changes in DNA methylation and hydroxymethylation. We can observe loss of imprinting. For example, some genes should be silent on maternal or paternal allele, and they are in all normal cells of our body. In cancer cells, we lose this imprinting, so both alleles will be expressed. So this is observed, for example, for this gene and some cancer types. Also, we often observe hypomethylation of CpG island in some important genes, for example, tumor suppressor genes. And although often we observe accumulation of CpG methylation on CpG islands, we also observe hypomethylation of genomic DNA overall the genome. So we lose methylation everywhere, but we gain it on specific CpG islands in cancer. And often we can use information about this methylation of promoters to stratify patients into patients with bad prognosis and good prognosis. Here I show you 2 types of classification. Here, both for skin cancer, And here, based on the methylation of promoter of estrogen receptor alpha gene, and here it's another gene. And if you have the promoter of the gene methylated, the survival is really bad, goes to 0. And if you don't have the promoter of the gene methylated, the survival is much better. So you can use information about DNA methylation to say this patient has a more aggressive disease, you must treat it in a more aggressive way, and another way around.
Probably for estrogen receptor, you usually don't use methylation to find out if it's expressed or not. You know, you usually do some it's like psychological screen rather than methylation. So I guess here is rather reflects the fact that the receptors is expressed or not. Yeah.
So sometimes, I don't know about this particular case, but I know that methylation can help you to select treatment. For example, you know that patients who have BRCA1 mutated, they get special treatment for breast cancer. Actually, the same treatment was shown to be effective if you have methylation of the promoter. If you know just the level of expression of this gene, it's not enough. You need to know that gene is mutated or promoter is methylated to apply this treatment. So I don't know for this special case what it works. Also, another example. So here it's for adrenocortical carcinomas, and we can separate tumors of this type into two subgroups. One group has a hypermethylation of about 500 promoters, and this phenotype is called CpG methylated phenotype. Another group doesn't have this hypermethylation, and you see again that the group with this hypermethylation has much worse survival. All right. Also, what we see sometimes in tumors is that we gain this CpG methylation not just for one gene, but for many genes in a row, more than 10 genes. And this phenomenon is called long-range epigenetic silencing. Often, when we have several tumor suppressor genes located close to another, we can silence them off by applying CpG methylation everywhere in the region, or polycomb repression. And this is an example of this long-range epigenetic silencing in the HOX-D cluster. And here I don't show you DNA methylation, but H3K27 trimethylation. It's again a breast mark which correlates with the CpG methylation. And you see that the whole gene cluster is silent by epigenetic mechanisms.
Yeah. Is it somehow related to that? Is it a single TAT?
Is it the same TAT? Maybe, yeah, but I didn't check. But you may be right, this is the same TAT. Another type of epigenetic modifications that we observe take place in cancer is loss of LOX, loss of large organized chromatin lysine modifications. This is lysine 2. And now we know that lysine 9 dimethylation is associated with LUDs. So what we see in cancer, that we lose this LUD marker. It means that in cancer cells, regions that should be attached to lamina are not attached to the lamina anymore. And genes which should be repressed because they are attached to lamina become reactivated. So you can refer to these publications. Another phenomenon which we observe taking place in cancer cells is de novo creation of enhancers and super enhancers. First, what's the difference between typical enhancer and super enhancer? Typical enhancer is just normal enhancer where we have a couple of transcription factors that bind. It's a small region, can be upstream of the gene and downstream of the gene. And a super enhancer, it's a very long region, more than 20 kb generally. And we have many different transcription factors that bind there. We have mediator protein that binds there. You have also H3K27 acetylation that covers the whole region. And if you look at the score for each super enhancer and gene expression in log scale, you see that it's almost linear. The stronger the super enhancer, the higher the gene expression. And if you want to overexpress a gene, the logical way is to create a super enhancer huge active regulatory region that can include the gene. And then the expression will go to the sky. And what we see that in some cancers, for example, colorectal cancer here, de novo superenhancers are created to activate genes that correspond to hallmarks of cancer. If you look at some superenhancer literature, you will often see this type of plots. On the x-axis, you see enhancers and superenhancers ranked by HVK27 signal. The stronger the signal, the higher the chance that it's a superenhancer. And the y-axis, they're just these regions. Do you have a question?
Is there a conservation between de novo superenhancers and different cancers and their positions? I mean, what genes they activate? Is that a hard question?
This question has not been studied really well yet, but I think you can answer it. So here I just want to show that for this particular cancer type, you see activation of invasion and metastasis by creating a superenhancer in front of this gene. You have just a superenhancer that which is created close to a gene which is responsible for sustaining proliferative signaling, inducing angiogenesis, tumor promoting inflammation, genome instability mutation, and so on. And how you create these superenhancers? Nobody knows. There are some examples where we know that some genetic event resulted in this epigenetic event, creation of superenhancers, but in many cases we just don't know. I will give you 3 examples where we know what happens. For example, in T-cell acute lymphoblastic leukemia, we observe somatic mutation upstream of tau1 gene, and this somatic mutation, short one, creates a new binding motif for the meat transcription factor. Meat binds there, creates a superenhancer, and the expression of tau1 gene, tau1 oncogene, goes to the sky. There is another phenomenon which is called enhancer hijacking. It's been already described for neuroblastoma and medulloblastoma. In neuroblastoma, the TERT gene, which codes for telomerase, gets an active enhancer through a translocation. So there was an enhancer or superenhancer somewhere quite far away from TERT. Because of a genetic event, translocation, this superenhancer was put close to the promoter region of TERT, and TERT was activated. And we know that if you have active TERT, you have lots of telomerase, and then the cell can divide non-stop. In medulloblastoma, the same type of mechanism was observed to activate the gene, oncogene family GFI1. But again,
By hijacking you mean that it's just change of 3D structure? Or
No.
It's like
No.
Oh,
It's a translocation, yeah. But in many cases, we observe this superenhancer there. We have no idea why it's been created. In all normal cells, there is nothing like superenhancer in this region. In a cancer cell, there is one. And you can use information about the superenhancers to provide, to suggest treatment. Because there are many epigenetic treatments, some of them disrupt the action of superenhancers. For example, inhibitors of CDK7 BRD4 proteins decrease significantly expression of genes that have superenhancers. And here it's a work by the group of Ronnie George, where they say that neuroblastoma cell lines that have MECAN amplification have superenhancers in these 4 important genes, ALK, COX2B, DBH, GATA2. And this is why cell lines that have this MECAN amplification and thus have these superenhancers, they are sensitive to CDK7. CDK7, again, it's an inhibitor of the action of superenhancers. And indeed, if you look at the cellular survival, cells that have this MECAN amplification, they die much quickly than cells shown in blue that do not have this superenhancer. And I must tell you from a clinical point of view, neuroblastoma patients that have this MECAN amplification, they can be hardly treated. More than 60% of them die. Using this treatment is a hope for these patients. And this treatment can be suggested because this epigenetic analysis has been done. And this is just to show that in mouse, it works as well. All right. I told you about changes in the epigenetic landscape when we see some changes in histone modifications, but also sometimes we see mutations or changes in histone variants. For example, in many cases we see mutations in ATRX gene or DIX gene or histone variant 8.3. All these changes result in alternative lengthening of telomeres, which is called out. So here you see normal nucleus and you see that telomeres are quite short. And if you have this alternative lengthening of telomeres mechanism on, you see that telomeres become really, really long. And for cancer cell, it's very good to have these long telomeres because it can divide and divide non-stop. So mutations in this gene can result in this phenomenon. And also, if you have it, you have more breaks in chromosomes. So it's quite difficult to read this picture, but just believe me, if you have these genes mutated, in addition to this lengthening of telomeres, you have much more copy number alterations. And copy number alteration when you have a loss or a gain of chromosome arm. And in cancers, we would like to lose and gain chromosome arms because some chromosome arms contain lots of tumor suppressor genes. For example, 17p arm contains lots of tumor suppressor genes. And some chromosome arms contain lots of oncogenes, 17q, so we want to amplify them. All right, so this is a summary for the first part of my talk. I told you that chromatin is organized in LAD and TAD, and this LAD and TAD structure is related to histone modifications. To identify them, you can use DMID, Hi-C, 5C, and so on. Histone modification and histone variants have direct effect on transcription. They form groups and indicate distinct chromatin states. To profile these chromatin states, you can use the ChIP-seq and then apply FigMark models on the ChIP-seq signals. Also, epigenetic states change in cancer compared to normal cells, and we observe more than 30 different genes coding for chromatin remodeler genes, proteins mutated in cancer. And information about epigenetic profiles can be used to stratify patients and propose some epigenetic treatments. In cancer cells, we observe loss of imprinting, hypomethylation of CpG islands on promoters of many genes, including suppressor genes, genome-wide DNA hypomethylation, long-range epigenetic silencing when I have many genes in a row silent, loss of large organized chromatin lysine modifications, LOX, and thus this LAD structure can change, disruption of TAD LAD, depletion of histone variant 3.3, and thus alternative lengthening of telomeres, and creation of de novo enhancers and super enhancers. And maybe I should stop here and not go for the second part of my talk. Why?
No, yeah, fine. You start talking about it later.
All right, then it's going to be short, the second part. Kids, what methods we use to analyze ChIP-seq data in cancer cells. And the challenge here is that we analyze cancer data. And cancer cells and cancer DNA, it's not the same as normal DNA. In cancer cells, we have copy number alterations. So here I show you 24-color karyotype for lung adenocarcinoma cell line. Each chromosome has its own copy. So you can see how many chromosomes 1, 2, 3, and so on you have. For example, you have 2 copies of chromosome 10, 1 copy of chromosome 6, 3 copies of chromosome 7, 4 copies of chromosome 5. In addition to these duplications of losses of chromosomes, you have translocation. For example, you see pieces of yellow here, and yellow may be chromosome 1. So we have a translocation between chromosome 4 and chromosome one.
It's a cell line or primary sample?
It's a cell line.
But cell lines are even worse than primary samples.
It depends on tumor type.
Always.
Maybe. And if you apply existing techniques to find regions which are enriched in particular histone modification, you will see that these techniques will give you much more signal in the region of gain compared to the region of loss. So here I show you chromosome 8. This part was lost, this one was duplicated, and this is regions of enrichment of HBK27 translation predicted by Max or Sicer. These are two tools used by ENCODE consortium to predict regions of enrichment of histones. And you see that it misses lots of signal in the region of loss. Thus, we developed our method to correct for this copy number, which is called HMCAN for histone modification in cancer. It's fully available online, published in weblinks. And the principle is simple. We correct for different biases, including copy number bias. And to get information about copy number, we take an input sample. Input, it's a control for ChIP-seq when we just cut DNA and we sequence it. And we use it to find regions which are enriched in reads, meaning gains, and regions of loss. And then we apply hidden Markov models, again, to segment our profile into regions which are enriched in chromatin marks and regions of background. And if you look at the output, you see that HMCAN does not have this copy number bias. So it can be applied on cancer cells. And this is a method to say this region has a mark, this region is a background. Sometimes you want to compare two samples. For example, you want to compare a normal sample with a cancer sample. So you want to find differential enrichment. And for this, we developed another method which is called HMCAN-DIF, which has been submitted to a journal. But anyway, it's available online. And here again, we do normalization for different biases, for GC content bias, copy number bias, library size bias, so on. And then we apply hidden Markov models to predict three states. No equal enrichment, high enrichment in one condition, high enrichment in another condition. And apparently works really well. This is validation on simulated data and comparison with other methods. On the left, you see precision recall curves when we compare two samples that do not have copy number alterations between them. For example, we compare a normal sample versus normal sample. And you see that recall and precision are very high for all methods, including our method, which is shown in light blue. So everything works. If you work with normal data, take any method, it will do the job. However, if you want to compare a normal sample with a cancer sample, then immediately we see that all existing methods just fail. They don't find the signal and they give lots of noise. Our method is closer to the ideal 0.11. It's not perfect still, but at least it works much better. So if you have cancer data, ChIP-seq data, I would suggest that you use our method. Most of the tools we developed to analyze ChIP-seq data are also implemented in the Nebula ChIP-seq analysis framework. So you can go to nebula.q.fr, and here you can see a Galaxy-based platform to work with your ChIP-seq data. You can align your reads, find signal, do motif discovery in the regions of interest, compare two signals, annotate them to say that this signal is found in the promoter of the gene or this one enhancer and so on. And it will produce the useful graphs that you can then include in your publication. And this is all. So I will just summarize the second part. If you want to analyze your epigenetic profiles of cancer cells, you should do it using different techniques, high-seq, ChIP-seq, DNase, high resistivity, ASIS, and so on. But you should keep in mind that you have these copy number alterations in cancer cells, and the method you use should take it into account. And also, one thing I didn't cover at all is that if you take a tumor sample, not a cell line, this tumor sample will be contaminated by normal cells. So the signal you will see will be a mixture of several signals. One will come from cancer cells and others will come from normal cells. And you need somehow to correct for it. And there is no method so far to decontaminate ChIP-6 signal when it comes from a primary tumor. All right, so this is all. You can ask questions in Russian, in English, French, if you wish.
Spanish.
You can at least ask.
I'm sorry for asking so many questions, but you know, in the field of methylation, there's this concept of partially methylated domains. So when you have a long range, like values of the methylation, and I'm wondering, like, what would they correspond to, like what you were you were telling? Are these logs, like when you lose connection to lamina?
I'm not so sure about it. Maybe not. Yulia, maybe you can help me.
Actually, I was about to ask the same question, but not about lactase-associated domains, but more about superenhancers.
Yeah,
Yeah.
it could be also super.
Yeah, actually, often we see these regions which correspond to regulatory elements, enhancers, and super-enhancers. Not only super-enhancers, but also enhancers. But I think this question has not been really well studied.
Yeah, that's why I'm asking. I'm just
But it
interested in your opinion.
It looks very attractive that you lose methylation in a very huge region, and because of the loss of the methylation, it can gain this kind of enhancer.
Yeah, maybe, yeah.
But do you see some specific features of super enhancers in terms of methylation? Like, imagine that you have only methylation data for some cancer, like which is the usual story. When you don't have any ChIP-6 data, only methylation data for cancer, can you actually study super enhancers with this data, or it's not enough?
There is a recent work that they show that you can actually annotate enhancers rather well using DNA methylation data. If your coverage is very good, you can see all CPGs, not with the array data, but with the bisulfite sequencing data. However, just one work, and they show that it can work, although why should you do it if you can do ChIP-6? It's not super expensive and combine the types of data. So in principle, maybe yes, but why would you do it?
Thank you.
Maybe that, so the essence of your methods that the new one is basically that you have spike-ins for different regions of the genome. That's the key.
No, we don't have spike-ins. It's a good thing to have spike-in DNA or chromatin
Yeah,
to renormalize the
Yeah.
signal.
but you don't.
We don't, although it's a good idea. We don't have it. We have not implemented it because very few labs do it. Everybody knows it helps. Nobody wants to pay more extra to do it. So what people do, they don't normalize at all. What we do, we say most of the regions should stay the same.
Like RLE normalization for them.
So what we call peaks, and we try to do kind of regression to say most of the regions should not change the signal.
Yeah.
And this is our normalization without any spike-ins.
There's some kind of median assumption.
Yeah, exactly. Yeah.
When it said about methylation status of cancer cells, is it about primary tumor? And what about methylation of cells, cancer cells, in metastatic sites, for example?
It's a very good question, and I don't have an answer for this. Do you know about the difference between metastasis and primary tumors and methylation?
And also, quite similar question, what about methylation status in tumor when it progresses, for example, during the different time periods?
I don't have, it's a very good question. I don't have the answer. I can tell you that for histone modifications, we are going to answer this question for neuroblastoma. So we got money to do 100 tumors, including primary tumor metastases or different time points. So we will check whether epigenetic landscape, meaning enhancers, super enhancers, polychrome repressive domains change during the tumor progression. So we will answer this question for histone marks, but not for DNA methylation. And I'm not aware about works about DNA methylation. They must exist. but unfortunately I'm not aware of it.
More questions? We finished.
Okay, there's no. So I guess now we will continue with Finn. Here's just a brief introduction. Finn is